Hello students, welcome to Konnur online classes, and today we are moving on to the next chapter, that is biomolecules. See, before entering into the chapter, we will discuss some important things. That is, as we can understand, imagine it is Earth. So, when we take the Earth, I mean to say that the whole locality, when we consider the whole locality is considered as Earth. So, when we take Earth, within this I can make some compartments. So, in this a small or one compartment or each compartment is considered as biosphere. Do remember? Yes. So, when we take the whole locality is considered as Earth. Within the Earth, when you take a small, one quantity or each part is considered as biosphere. So, the biosphere is nothing but it is a unit of Earth. Biosphere means it is a unit of Earth. So, in the biosphere, again we will be having many compartments. One is, as we know that atmosphere, as we know that hydrosphere, and as we know that is lithosphere, yes or no, and still many are there, as you know that is xerosphere, yes or no. So many are there. In that, when you take atmosphere, I mean to say that here we will be having air, yes or no. So consider as atmosphere, and hydrosphere nothing but you will be having water. Here you will be having air. The same time lithosphere nothing but here the soil. Yes or no? So, at the same time, here you will be having many microorganisms. Yes or no? So, these lightweight microorganisms can be wandering in the air. So, nothing but atmosphere, and water. Again, here you will be having aquatic animals, as you can understand. Yes or no? Most of the aquatic animals, I mean to say, so all the aquatic animals are present in the water. So, that particular area or particular habitat is considered as hydrosphere. The same time, when you take a soil, I mean to say all living system including human beings are present in on the earth crust is considered as lithosphere. So this is what I mean to say here when you talk about the biomolecules, and we all living organisms including microorganisms, lower organisms, higher organisms, animals, plants, etc. all are present on the earth. So here what I am trying to tell you is that, so when we people are in the biosphere, so those all living system, whatever you take, right from microorganisms to till higher organisms, so those all living systems are made up of at least one or the other chemicals. Very important point. Yes or no? Yes. So when you take all living organisms, so those all living organisms are at least made up of chemicals. So it may be same chemical or it may be different combinations of chemicals. So this is what I am trying to tell. So here when you take this lithosphere, as I told you people, here you will be having a living system. So including human beings, here again aquatic animals, again they are considered as animals, and sometimes weeds, nothing but algae, different plant bodies are there. Here microbes, so that microorganisms, animals including plants, again organisms, animals including plants. So that finally when we take all these, so here you will be having organisms. Yes, we will be having organisms, and here you will be having what? Animals. The same time you will be having the plants. Clear? And one more will be microorganisms or microbial paste, whatever you take. So these all things are made up of chemicals. My final conclusion is that, so these all organisms are present on the earth, these are made up of one or the other chemicals. I hope you understand. This is what the thing about the biomolecules. See, next is, as I told you right now, these all organisms, nothing but the better word is living system. Yes or no? You can take microorganisms, or you can take animals, plants, or microorganisms including viruses, whatever are there. So those all are made up of one or the other chemicals. This is my conclusion. But now what I am trying to tell you is that, so the thing is, these organisms, these living system are made up of same chemical or different chemicals. Clear my point? I mean to say that these all living system are made up of similar or same chemicals? Question mark. All organisms are made up of same chemicals or different chemicals. Say for example, see here I can take one living organism, that is animal. So take an example of tiger, and the same time I can take a plant, that is rose plant. One thing, that is both come under living system. Yes or no? So this animal, that is tiger, as well as rose, these two come under living system. But my question is that this tiger and rose both made up of same chemical compounds, same elements, same molecules, or different chemicals or different compounds or different elements. This is what my question. So that I am going to put question mark. Clear my point? So that this is what we need to understand. All living organisms are made up of same chemical or different chemicals. See, and when you start searching whether all living organisms are made up of same chemical or different chemical, so when we start searching this answer for this question, then finally we will get, I mean to say, some of the things are there to understand. One is, say for example, I can tell you people, that is hydrogen, or you can take carbon. Better word is carbon. You can take. Yes or no? So when you understand these two molecules, or we can take these two molecules, that is hydrogen and carbon. At the same time, when we take an animal, the same time when you take a spoon of soil, so that same time, meanwhile we are going to get the hydrogen in the animal body. Am I right or wrong? Yes, definitely. So that when you take animal body and when you take the chemicals or chemicals present within the animals, definitely we are going to get hydrogen. Yes or no? So hydrogen must be present in the animal body, including my body. Yes or no? So same time, when we understand, or when you take out, or when you analyze the chemicals present in the soil, no doubt definitely you are going to get hydrogen in the soil. Yes or no? We can take it as a earth crust. Earth crust. I mean to say that hydrogen, I mean to say the same molecule or same element is present in the animal body, and the same element is present in the soil. Yes or no? At the same time, when you take a carbon as an element, this element of course is present in the animal body, and the same carbon is present in the earth crust or in the soil. Yes or no? So for that reason, and my question is that, see, the same hydrogen is present in the animal body, the same time same hydrogen is present in the earth crust. Are they same? Are they same? Yes, at the same time, carbon, the same carbon is present in the animal body, the same carbon is present in the soil. Again, are they same? So this is my question. Yes or no? So think about this one. So for that reason, my final conclusion is that no doubt the hydrogen is present in the animal and soil, and the carbon is present in the animal and soil. No doubt the element will be remain same, but their functional aspect, you must remember this, their functional aspect will be differ. Yes or no? Functional aspect will be differ. So one better example I can give you people. So when you take a carbon, that is C6H12O6. Just imagine C6H12O6. Definitely, as you know this is considered as a glucose molecule. Yes or no? So when you take this glucose, in the glucose, here you will be having 6 carbon atoms are there. Yes or no? There are 6 carbon atoms are there. So same carbon, as I told you right now. So that the same carbon atoms, I mean the 6 carbon atoms are present within the animal body. Yes or no? So they are present in the animal body. At the same time, when you take SiC, called as silicon carbide, as you can understand according to the chemistry, silicon carbide, it is present in the sand. Yes or no? So here we will be having, when we consider this glucose and we consider this silicon carbide, here both, I mean the glucose contain carbon, the same time silicon carbide contain carbon, but their functional aspect will be differ. Yes or no? So here it play a very important role in the providing the energy to the body, but it is not playing any role in the energy. So just it is going to bind with the silicon. Yes or no? So but their their functional aspect will be definitely, there will be a different. Clear? So this is what I am trying to tell you people. Clear? This is one concept. At the same time, just a minute. Yes. Next thing is, by understanding all these things, finally we came to a conclusion is that no doubt the living organism and non-living things, living things and non-living things. So most of the living and non-living things contain same molecule or same element, but thing is their functional aspect will be different. So that now the question is that what are the chemicals, what are the different chemicals present in the living system and non-living system? That is we need to understand. So my question is, what are the different chemicals, or when you take any sample or when you take any material that is living material, so that contain what are different chemicals? Yes, so to understand what are the chemicals present in the material, we will be having an experiment called as chemical analysis. So do remember, in the examination they may ask chemical analysis, a small experiment to understand what are the different chemicals present in the given sample. So while conducting this experiment, what are the things we need? First one is, we need biological sample. First one is, we need biological sample. So now we can ask, sir, what mean by biological sample? Yes or no? So here the biological sample means that sample which is collected by the living system. Clear my point? So the biological sample refers to the sample which is collected by the living system. So here I can write some of the example from plant source. First I can write the sample which is collected from the plants, such as leaf. Yes or no? Definitely leaf is considered as a living system, so it can come under biological sample. You can take flower, you can take stem, you can take root, you can take bud, you can take any other part of the plant. That's simply considered as a biological sample. When you take animal source, here we will be having what? Skin, you will be having liver sample, yes or no, and spleen, kidney, whatever the organ you take. So not considered as biological sample. The same time, when you take sample, nothing but imagine, it is what? Biological sample. Imagine biological sample, I can write one will be plant source, another one will be liver source. So better I can take liver. Yes, so we can take the liver from the dead animal. Do remember, dead animal. So from the dead animal we need to take the liver, and simultaneously we need to take what? You can understand this. Yes, we can write it as a mortar. Is mortar. So here put that liver, that is biological sample. I mean to say, take that biological sample in the mortar, and the same time here you will be having what? Yes, it is considered as pistol. Yes, I mean to say like a grinder, so that when you take the mortar, in the mortar when you take the biological sample, it is considered as a lever. I mean to say biological sample. The same time when you take pistol, and very important thing is that to this we are going to add, that is Cl3CCOOH. Yes, can you? What is this? It is simply considered as trichloroacetic acid. Do remember, trichloroacetic acid. So simply consider as Cl3CCOOH. So by addition of this chemical to this mortar and try to grind the biological sample. So try to mince it as much as possible, try to mince it. So once it will completely mince, now you should take after complete grinding. So try to grind it. After complete grinding, take a test tube. Very important concept. See, imagine it is test tube. Yes. Now, very important thing to understand is whatever the chemicals are present here, I mean to say, so whatever after complete mince, after complete grinding, whatever the chemicals or the things are present in the mortar after addition of trichloroacetic acid. So now cover this test tube with it is considered as cheesecloth. Do remember, it is considered as cheesecloth. If I mean instead of cheesecloth we can use cotton, no doubt, no problem. Yes, there are two the cloth, I mean to say like a filter, we can call it as a filter. So one is a cheesecloth, second one is cotton. So after putting this cheesecloth on the test tube, try to pour all the content which is present in the mortar like a tea. Yes or no? So after pouring, we are going to get two fractions. We are going to get two fractions. One is whatever the fraction is present, I mean which is filtered. Yes or no? So it's considered as filtrate. Please do remember, it is considered as filtrate. I mean to say whatever we get in the test tube after pouring, we are considered as a filtrate, and this is also considered as acid soluble fraction. It is also called as acid soluble fraction. In the same time, whatever the content is retained on the cheesecloth, even after pouring, whatever the content is present on the cheesecloth, which is held by the cheesecloth, is considered as what you can call it as, that is filtrate. Here the word is retentate. We can call it as retentate, and this retentate is also called as acid soluble, and here we can call it as acid insoluble. Do remember, also called as acid insoluble, and one is acid soluble. And what we get in the test tube is considered as acid soluble, and what is remain present on the test tube is considered as acid insoluble fraction. Acid insoluble fraction. Clear this? Got an idea? Yes or no? Here test tube. In the test tube, one will be filtrate called as acid soluble, and one will be retentate also called as acid insoluble. And our scientists have discovered the more than thousands, do remember, more than thousands organic, do remember this word, our scientists have discovered more than 1,000 compounds, more than 1,000 compounds in the acid soluble fraction. Clear this? Our scientists have discovered more than 1,000 organic compounds present in the acid soluble fraction or in the filtrate. Clear? So how? Now you can ask a question. So how? What is the, what are different various techniques? Various separation techniques. Do you remember? These all 1000 different organic compounds has been categorized or isolated by several, do you remember the point? Several separation techniques. Yes, where there are various or several various separation techniques. As you can understand, I can tell you some separation techniques. One is column chromatography, then thin layer chromatography, paper chromatography. Yes or no? So, and one more is definitely considered as chromatography. By means of all those different techniques, we can separate or we can isolate the thousands of organic compounds. Clear my point? This is one topic regarding the biomolecules, and by understanding these all different, is finally whatever the 1000 different organic compounds what we got from this experiment called as chemical analysis experiment, finally we can write the structure. Is according to the chemistry, whatever the organic compounds or whatever the chemicals present, so definitely we need to write their structure. Yes or no? And one will be their formula, their chemical formula, and one will be their structure, chemical structure, and one will be chemical formula. So we need to write these two, that is, one will structure, another one will be formula. We can by means of that IUPAC a name, whatever. So, and by understanding this all topic, finally I can conclude, I can write the definition. What is biomolecule, or what are biomolecules? What mean by biomolecules? Yes or no? So by understanding these all things, definitely I can write what is biomolecule. So all carbon compounds, note it, all carbon compounds, what we get from where? Definitely from the living system. Yes or no? So all carbon compounds what we get from the living system are simply considered as biomolecules. Do remember? Clear my point? So finally we came to a conclusion that all carbon compounds what we get from the living system are simply considered as biomolecules. Again I will come back to the topic, that is earlier I told you people, that is the carbon which is present in the soil, same time the same carbon is present in the animal body or in the living system. Are they similar? Now you can give the answer. Yes, is it possible to give the answer? That is, the carbon present in the soil, carbon present in the animal body or in the plant body or in the microbes, are they similar? Yes, I asked a question, that is whether they are similar or they are dissimilar. So now you are in a position to answer the question. Yes, are they same? No, because yes, according to the definition, the carbon compounds, all carbon compounds what we get from living system. A very important point to understand, that is living system. That the carbon present in the soil, same carbon present in the living system. No doubt both are carbon, but thing is, but our definition is going to tell the carbon must be extracted from the living system. Do remember this point, so that the same carbon is present in the soil, it is not taken out from the living system, it is from the non-living system, so that they are not similar, they are dissimilar. No doubt they are same carbon, they are same element, but they are different. I told you people that their chemical aspect will be differ. This is my conclusion. I hope you understand this. This is what one topic regarding the biomolecules. By understanding these all, finally we came to a conclusion, or we can we can frame a sentence or the definition, that is all carbons compounds are, that is what we get from the living system are considered as biomolecules. Clear? And at the same time we will be having one more small experiment. Yes, I hope you people are following my points. Yes, okay, we'll have a small topic, that is again I'm going to ask you people a small question, that is, as I told you, by conducting this experiment called as chemical analysis. So by conducting the chemical analysis experiment, finally we came to a conclusion that is our scientists have discovered 1,000 different organic compounds. Yes or no? But now my question is that, so here it contain only the organic compounds? It contain only organic compounds? No. Along with the organic compounds, it also contain, I mean to say that filtrate, I mean to say acid soluble fraction, and one more is acid insoluble fraction. So both contain not only the organic compounds, they also contain inorganic elements, inorganic compounds, and inorganic molecules. There are three things. One is, I mean, organic compounds, inorganic molecules, inorganic elements, inorganic compounds. So, and how to understand and how to separate and how to categorize the inorganic compounds? Yes or no? So that this is a one such experiment called as chemical analysis to separate organic compounds, and we have one more experiment to isolate, that is inorganic elements and compounds. So they are called as destructive experiment, called as destructive experiment. Very simple experiment to understand. That is what to do here. Again, in the first step we need to take biological sample. Yes, as I told you people, what mean by biological sample? The sample which is collected by the living system, called as biological sample. So again I can take some examples, that is from plant source leaf, and from animal source, that is liver. Clear? So consider as biological sample. So when you take the biological sample, after selecting the biological sample, I mean to say which sample we need to take for the experiment. See, after collecting or after selecting the biological sample and allow it to dry. Allow it to dry. So what happens when you allow it for drying? Within a very span of time, all water will be evaporate out. Am I right or wrong? So definitely after certain time, all water can be evaporate out, so that once all the water can be removed out from the biological sample, so definitely what we get, that is same biological sample, but it is completely drying. Yes or no? Completely dry sample, we can call it as dry sample. So now this biological sample or dried sample can be completely burned. Completely burned. Clear this? So when you take a biological sample, allow it to dry. Allow it to dry means remove complete water from it. So when we remove the complete water from it, definitely we'll get the complete dried biological sample. That dried biological sample can be burned completely. So in this, when you burn it, you're going to get two fractions. Again, whatever the carbon compounds are there, please do remember, whatever the carbon compounds are present in the biological sample, those all can be oxidized. Those all can be oxidized in the form of carbon dioxide or water vapor. Do remember, very important. See the question. So when we burnt the biological sample, whatever the carbon compounds are present in the biological sample, those can be oxidized in the form of carbon dioxide and water. At the same time, what we get is called as ash. That ash contain inorganic elements, inorganic compounds, and inorganic molecules. Clear my point? This is what our second experiment. See, why the and Why we allowed the carbon to oxidize? Because all those carbon compounds we already got in the first experiment called as chemical analysis, and the intention of the second experiment, that is destructive experiment, is to isolate the inorganic elements, compounds and molecules. Again we will be having different or various separation techniques to isolate the inorganic elements, compounds and molecules, such as we can call it as calcium, magnesium, manganese, and different various are there. Yes, and remaining things we'll discuss in next class. Thank you.